Hello, we are the FlowerEye team representing the PSU Center for Pollinator Research. They are Penn State's lead researcher on pollinator populations, supported by the College of Agricultural Sciences and Huck Institutes of the Life Sciences. Their goal is to conduct and communicate research that advances the health of managed and wild pollinator populations and promotes the conservation of their ecosystem. Bees learn from visual floral stimuli and sucrose rewards, but manual observation of this learning process takes significant time and effort, as well as suffering from human error. The FlowerEye is a tool that automates the observation of bees for the sake of cognitive and behavioral research. For the project to match the scope of a semester-long capstone, our sponsor proposed that our project build upon a pre-existing open-source model called the Insect Detect. Our objective was to create the model that improves upon the Insect Detect by opting for an LED platform with 2 adjustable flower patterns, as well as the addition of an automated microfluidic system to simulate nectar rewards. In the early stages of the project, our primary focus was on replicating the core functionality of the Insect Detect system. This involved designing a physical enclosure in CAD and ensuring that we could reliably capture an overhead video of the platform area. After confirming that the camera system was reliable, we began adding interactive components. The first major addition was the LED matrix board, which serves as the visual stimulus platform. This allows us to simulate artificial flowers using programmable colors and patterns. We were able to successfully control the LED board using the ADA Matrix portal and display multiple patterns. Next, we began developing the microfluidic reward delivery system, which uses syringe-based reservoirs, tubing, and a peristaltic pump to deliver fluid to a designated area. By the final prototype stage, we were able to successfully control fluid delivery through a graphical user interface, GUI, running on the Raspberry Pi. This demonstrated that the system could not only deliver fluid, but also allow user-defined control over when the delivery occurs. The microfluidic delivery utilizes a Raspberry Pi to run a local Python interface, control 2 12-volt pumps through MOSFET switching, and use the syringes and tubing to route fluid to the top surface. Our LED visual stimulus is a 64 by 32 matrix portal S3 RGB LED panel that utilizes bee visible colors and simple flower-like patterns in our demo. In this version, the systems do not communicate, but are physically co-located. The tubing dispenses onto an acrylic surface directly above the LED panel, so the reward location lines up with the displayed flower queue. As it stands, the FlowerEye is a promising prototype for a bee cognition research tool. The LED patterns run on standalone power. The pump control path has been confirmed. The GUI can save presets, validate settings, and execute schedules cleanly. Safety behavior is also built into the code, with pump shutdown on the normal exit, stop, or execution. The next phase of the project would focus on refining and validating the system. First, improve reliability by replacing temporary wiring with permanent connections and mounting components securely within the project box. Second, quantitative testing the microfluidic system to ensure accurate and repeatable fluid delivery. Third, integrate computer vision detection into the system by connecting the camera feed to a detection algorithm. Finally, further improvements to the LED system, enclosure designs, and overall usability will help transition the prototype from a functional demonstration to a more reliable experimental platform.